All right, it is now 7 o'clock and we're going to go ahead and get started. Thank you to everyone for joining us this evening. This evening, we're going to explore some amazing cancer research at Virginia Tech. Hokies are well known for taking on the state and the world's most pressing challenges. But people might be surprised to know what a force we have become in the fight against cancer. More than 30 research teams are trying to solve problems associated with this terrible disease. We are going to take a look at just a tiny slice of that exciting work this evening. Let's turn our attention to Dr. Michael Freelander, who will start this conversation for us. Dr. Freelander moved to Roanoke from his neuroscience leadership role at Baylor College of Medicine in Houston with his wife, Sandra, in 2010 to become the inaugural executive director of the Fralin Biomedical Research Institute at VTC. In addition to serving as the Institute's executive director and the senior dean for research at the VTC School of Medicine, Dr. Freelander was named Virginia Tech's first vice president for health sciences and technology in 2016 to spearhead biomedical research across the university and to lead the development of the VTC Health Sciences and Technology campus here in Roanoke. Among his honors and recognitions, Dr. Freelander is an elected fellow of the world's largest scientific society, the American Association for the Advancement of Science, a recipient of the William Meininger Award for Distinguished Contributions to Mental Health Research, and a distinguished scientist of the Society for Experimental Biology and Medicine. Dr. Freelander recruited some of the world's most creative biomedical scientists to Roanoke, leading the city's storied transformation from train town to brain town. He helped lead the efforts to develop the university's COVID-19 testing capability. About a year ago, he announced the university's cancer strategy. Dr. Freelander, would you take it from here?
Absolutely, Kim. Thank you very much. I appreciate your kind introduction. And I want to remind everybody, we all had our masks on, but we're sitting alone in our offices and have now taken them down. So it's a pleasure to be here with everybody this evening and to have the opportunity to share some of the exciting things going on at Virginia Tech, particularly in the cancer research space. And I have with me, who I'll introduce in just a moment, three really outstanding guests who will really carry the bulk of the evening. But let me start by saying just a couple things about what led up to the initiative that Kim just described that we're going to hear about tonight. So Virginia Tech, everybody knows, is a terrific university, world famous reputation in a variety of areas. Most notably, over many years, it's great strengths in engineering across the board, and a lot of other strengths in the life sciences, the social sciences, et cetera. But one of the things that I noticed when I got here, once we launched the Health Sciences Campus and the Fralin Biomedical Research Institute, is there didn't seem to be enough opportunities to bring together across the entire campus people with a certain focus and interest and passion around particular areas in the health sciences. And one of those, of course, is cancer. And I'm sure it comes as no surprise to anybody that cancer is a major and leading cause of death in the United States and in the world. In the United States, the latest statistics actually put cancer a little bit behind heart disease in terms of mortality, but it's rapidly advancing. And actually, in some states in the United States, cancer is the #1 cause of mortality and likely to keep advancing on heart disease, with a lot of the gains being made in treating heart disease these days. So it's a very important area of our health. Everybody's interested in healthy environment, healthy nutrition, healthy lifestyle, how those things might or might not impact getting cancer. And so there's a lot of interest in the community at large. Beyond that, at Virginia Tech, there's a tremendous amount of interest and expertise, both from a technological perspective and a conceptual perspective, that really comes from every corner of the university, all the different colleges, many of the departments, the institutes, et cetera. And so again, looking around, it became pretty apparent to a lot of us, and I simply acted as a facilitator to try to bring some of these things together, that there was a need and an opportunity for Virginia Tech to bring together its cancer research community. So we formed what we now call the Virginia Tech Cancer Research Alliance. And that alliance started with about 33 or 34 members from Blacksburg and Roanoke. And one of the interesting things I found out right away as we started to talk to each other is a large number of those investigators had funding for the research from what's called the National Cancer Institute, or NCI. It's one of the institutes at the National Institutes of Health, or NIH. And you normally wouldn't think we would have a lot of those because mostly those grants, which are very competitive to get, I should add, go to places that have large cancer centers, which we don't have. But we have a lot of individuals doing great work in cancer research. So we did an accounting of that. We brought people together, and now we've started to really move. COVID hit very early on this and gave us a little bit of a delay, but we're back in high gear again. So what we'd like to do tonight is I picked 3 representatives of the many that I told you about from this Cancer Research Alliance at Virginia Tech, and we're going to have a conversation with them. I've got some questions prepared that I'll ask them, but you're more than welcome, the audience, to ask questions if you'd like as well. We have a Q&A section, so please feel free to enter your questions in the Q&A. I'll take a look at those as the moderator, and I'll try to stop periodically and pick out those questions and throw them to the panel as well. So without further ado, I'd like to introduce the 3 members of our panel. Firstly, at least on my screen in the upper left, is Dr. Carla Finkelstein. Dr. Finkelstein is an associate professor of biological sciences in the College of Science at Virginia Tech, and she's located here at the Fralin Biomedical Research Institute in Roanoke on the Health Science campus. Many of you, I'm sure, have heard about Dr. Finkelstein's work with the COVID testing, molecular diagnostics lab, but perhaps you haven't heard as much about her research in cancer, and you're going to hear more about that this evening. She works particularly on breast cancer and is interested in what are called circadian rhythms and how that relates to cancer. So more to come in a moment. Hi, Carla, welcome, and we'll get with you in just a minute. The second person on my screen is Dr. Jennifer Munson. Dr. Munson is also located here at the Fralin Biomedical Research Institute on the Health Science campus in Roanoke. She is an associate professor in the Department of Biomedical Engineering and Mechanics in the College of Engineering. And Dr. Munson is very interested in sort of the mechanics of what's called interstitial flow or fluid flow within living tissues, and particularly in the brain, but other tissues as well, and how that plays a role in cancer. And she has a particular focus on a very malignant type of a brain cancer called glioblastoma, and how this flow works and contributes to that. So we'll hear more in a moment from Jenny. Welcome. Hi, Jenny. And then finally, our third, but last but not least, panelist is Dr. Eli Valessa Volkovic. And Eli is also an engineer in the Department of Biomedical Engineering and Mechanics, also in the College of Engineering, located in his case on the Blacksburg campus. And he has a particular interest in therapeutic innovations using a technology we'll hear more about called focus ultrasound and something called Hyssotripsy, and how you can actually use that to get at certain cancers and tumors and treat them. And he also has some great relationships with folks in the College of Veterinary Medicine at Virginia Tech and bringing our cancer team together. So these are the 3 panelists. And what we're going to do now is just I'll start with an individual question to each one and throw it out to them to get the conversation going. And as I said, as you think of questions in the audience, feel free to enter them in the Q&A, and then we'll go from there. So great. So Carla, we're going to start with you, if that's okay. So the first question I'd like to ask you to talk about, Carla, is what do you think the biggest challenge to the research community nowadays is to identify new and more powerful tools to prevent or treat breast cancer? And you know, can you tell us, is the need really for better diagnostics, new therapeutic strategies, lifestyle changes? Where do we really need to be focusing? Where's the greatest opportunity and need?
Sure. Well, thanks, Mike, for your introduction, and thanks my colleagues for being here and the audience as well. I think it's a very important question, and I think my colleagues would agree that independently of the type of cancer, the 2 major issues that we need to address are how to reduce the incidence of cancer, name it breast cancer or any other type of cancer, and how to reduce the cancer death rate. And I think in the United States, we have done really well in terms of reducing the overall cancer death rate. It has been steady declining since 1990. In the case of breast cancer in particular, we know now that 64% of breast cancer cases that they are diagnosed at a localized stage, meaning before the cancer spreads any other part of the body, okay, gives the patient 99% chances of survival, okay? Now, we're doing really well on that aspect. I think we can do way much better, and we can discuss the therapeutic approaches for that. I think we haven't done such a good job in cancer incidence. I mean, if you compare the statistics between 1950s and today, they're really side by side. I mean, it looks like we haven't done any job really in reducing the incidence of cancer in general. Now we do it better with some cancers than other ones. Now, why is that? Well, there are some risk factors that are associated with each cancer. In the case of breast cancer, for example, getting older, obviously, genetic mutations, reproductive history are all problems associated with developing breast cancer. You know, early menstrual periods before age of 12 is one of the issues. Personal history of breast cancer, those kind of things are difficult to somehow ameliorate. But there are other ones, there are other risk factors that we can certainly control and reduce the incidence of breast cancer. For example, being more physically active and not being overweight or obese. Now, in the case that particularly pertain to our research is controlling your circadian rhythms. We know nowadays that women that are exposed to abnormal circadian rhythms, let's say because they work at night or they have a misalignment, they are somehow off between their sleep and awake cycle, they actually have a 30% more incidence of breast cancer. And this is something that we can control.
Great, thank you. Thank you very much, Carla. It's a really good introduction to that topic, and I'm sure we'll hear more about the circadian rhythm aspect of breast cancer as we go forward. So let me turn now to Dr. Munson. Jenny, although malignant primary brain tumors, such as what's called glioblastoma multiforme, aren't nearly as common as many other cancers, like things we just heard about, like breast cancer, for example, lung cancer, prostate cancer, they have a real reputation for being particularly devastating. Why is that? What is it about these brain tumors?
Yeah, thanks, Mike, and good evening, everybody. So glioblastoma has quite a reputation. There's a lot of prominent people that we know who have died from this disease. And one of the things about it is that it is incredibly deadly to those people in terms of, though very few people get it, the 5-year survival rate or the number of people or the percent of people surviving after 5 years with this disease is around 5%. Which is extremely low compared to other cancers that some of which Carla just spoke about. In addition to this, those patients who are diagnosed with this often only survive around 7 to 13 months, the majority of those patients. So not only is it highly deadly, but it moves very quickly once it's diagnosed. And we think there's a lot of reasons for this. One is that it's very resistant to treatments. And though a treatment is quite aggressive, including surgery as well as chemotherapy and radiation therapy, these treatments just don't work, and these tumors do recur. And so that's why we see such bad death rates. And so another really devastating thing about this is that it really hasn't gotten any better in the last several decades. And so we really need novel treatments, novel targets, and novel ways to go after this disease so that we can really prevent it from being so deadly in these contexts.
Thank you, Jenny. Okay, Eli, let's turn to you. You're pioneering some very exciting new technologies that kind of blast or blow up tumors inside the living body. And there's a lot of interest in these kind of things, but I think in general, a lot of folks who don't work in this field are wondering, how the heck does that work? Does it work just by blowing up a tumor into a lot of small bits, or does that get rid of entirely? Or are you activating other processes in the body? What's behind these approaches using an energy like, say, focused ultrasound?
Yeah, thanks. That's a great question. So in general, focused ultrasound, or what we do is particularly the non-thermal type of focused ultrasound, which is called Histotripsy. So we're developing Hyssotripsy, and it's a completely non-invasive alternative to doing surgery or some minimally invasive ablation methods. So Hyssotripsy is a non-invasive, non-thermal, and image-guided focused ultrasound ablation method. And the way this works is we actually precisely focus sound waves or focus sound waves down to a single point in the body. We use very short, high-pressure sound waves that when we reach a certain peak negative pressure that only occurs at the focus of our transducer, we generate what's called cavitation, or a cavitation bubble cloud. And what these are is very localized, precise microbubbles that rapidly expand and collapse on the order of microseconds. And this rapid expansion of these bubbles exert very high stress and strain on the tissue that you're targeting. And so this basically disintegrates that tissue into an acellular debris. So basically what we're doing is we're precisely destroying the tumor. We're then scanning through a robotic positioning system to cover the entire tumor volume uniformly. so that we destroy not only the focal point, but all the tumor and the surrounding tissue we want to kill into this acellular debris with no remaining viable tumor cells. And this is basically, you're basically liquefying the tumor and you're left with, some, basically liquefied proteins, subcellular, but no viable tumor cells. And then over time, what we've seen in all of our preclinical studies and now our early clinical studies is that the body just naturally resorbs this. And we can talk more. I don't want to take up too much time on the kind of the immune response. And we have some great colleagues here that are looking at, we've actually seen that this can activate an anti-tumor immune response. But as a primary frontline therapy, we're basically non-invasively following this image-guided approach to just basically liquefy and completely remove the tissue. So this is still a really early technology. We've done a lot of preclinical work, and we've now moved into doing our first human clinical trial. This was done for treating liver cancer in Barcelona with our clinical colleagues there, led by Hyssotripsy, which is the industry leader in this. And now we're really looking at multiple applications, including some of the ones you mentioned, like moving into breast, brain, bone, pancreatic, soft tissue, and other tumors. So we really hope this can be a platform technology that's really focused on non-invasively doing what you can kind of accomplish with frontline surgery, but that's really limited to only a few patients who can get surgery currently. And obviously we hope to have much better quality of life with these non-invasive therapies.
Wow, it's exciting. So look, I'm going to take you up on what you just said. You said we can get into a little bit, and I think it's a natural flow. And I want to throw this question out to all of you or any of you. And so people have heard a lot lately, I think, about harnessing the power of one's own immune system and how that can be used to fight cancer. There have been some very promising new pharmaceuticals, new variety of new therapies that take advantage of this. So let me just ask a question that maybe a lot of folks in the public are asking. Is that the miracle cure that we've all been waiting for? And how does that work? And how do each of your approaches potentially interact with that? Or, any of the things you're working on in your research also tie into the activation of the immune system? Any one of you wants to start, go right ahead.
Yeah. Well, let me start, and then I pass the baton to my colleagues. Yeah, we asked that question as well, actually, Mike. We thought, what's the role here of the immune system? I mean, let me go back one step and explain what's the problem that we try to address. So we have, when you have a cancer cell, the cancer cell lives in a microenvironment. Imagine like an ecosystem, a bunch of things around the cancer cell. And this is a delicate balance. So the idea is that the immune cells that are around the tumor should eat that tumor and get rid of those cancer cells, but it doesn't happen. And that's one thing that has been driving us crazy for years, right? For decades, until finally, and 2 Nobel Prizes after, we discovered which ones are those checkpoint molecules that keep the immune system silent. And that's why the immune cells cannot eliminate the tumor. On top of that, we found that the way that the immune system is regulated around the tumor is regulated in a 24-hour cycle. What does it mean? It means that our normal immune system has a number of cells, and a particular number of cells at specific times of the day. What we found is that in the case of tumors, around the tumor, the number of cells and the type of cells varies at different times. And when that happens, if for any reason you have a person that has their clock completely desynchronized because has a chronic jet lag exposure, and when I say chronic jet lag, doesn't mean that it needs to take a plane. Just by working at night, you have the chronic jet lag. But when you're exposed to this chronic disruption of your circadian system, your immune system changes. And when your immune system changes, the tumor takes advantage of that and grows. So the question is, can we just help to control that immune system by going back and synchronizing that individual as part of the therapeutics? Obviously, it's not going to cure anybody, right? But it's going to be part of the therapeutic approach.
Yeah, thanks, Carla. Eli, Jenny, want to weigh in? Go ahead, Jenny.
Yeah, so in glioblastoma, therapies such as things like chimeric antigen receptor T cell therapies, or CAR T cells, as they're most commonly called, show a lot of promise, as well as for other cancers in the brain, such as metastatic melanoma or breast cancers. And so I do think there's a lot of promise there, but there's a lot of difficulties too. One is that accessing the brain for the immune system is quite tricky, because it is what's called an immune privileged organ, meaning that the immune system doesn't normally go there unless something's wrong. And so it can be very difficult to kind of direct these cells in there. And so how you deliver them, and as Carlos was saying, when you deliver them and what you're delivering, are all really important with these types of therapies. And so one thing my group looks at is how fluid flow can actually direct immune cells to move in and around tumors. And this is something that happens naturally in your body. If you get a cut on your finger or some sort of, injury, you have swelling at the site. And what's happening is that fluid and blood is rushing to that site, and then it's draining very rapidly. And it's actually helping those immune cells that get there to kind of get out of the infected area and then go to the places such as lymph nodes, where they need to actually tell the body how to mount that immune system. And so the same type of thing is happening during cancer, is you have enhanced fluid flow, and we may actually be able to leverage that or move it or direct it such that we can move these therapeutic T cells and other types of immune cells, and even the inherent immune cells in the body, in and around these tumors. And so that's one thing that we're trying to do, but it does take a lot of advanced knowledge of understanding what this fluid looks like and what these cells are doing and how they actually respond, and how can we help them by giving other therapies as well.
Thank you, Jenny. And Eli, I know you have some input with the therapies you're using and how they may interact with the immune system. Please.
Yeah, absolutely. And I'll start by saying I'm not an immunologist, and thankfully I work with, we have great collaborations here with Jenny and Coy Allen's group and others who are really focused on, characterizing the immune response after histotripsy and our other technologies. So I'm not going to talk too much about the mechanisms there, but I will say probably the most shocking thing in our research in the last 5 years, I developed devices where we're really focused on, show us where the tumor is and we will find a way to non-invasively remove it. And what we've seen is that these local therapies, especially our non-thermal therapies, so histotripsy, as well as there's other groups in our department here who develop irreversible electroporation. What we've seen with these non-thermal ablations, they actually activate an anti-tumor immune response. So we treat one tumor and we've seen abscopal effects where other tumors shrink in size, and we've seen synergistic effects when you combine them with some of these emerging immunotherapies, checkpoint inhibitors. So it's really exciting. Some of the mechanisms we think are involved is that we're actually breaking down the tumor cells, but we're not denaturing proteins. So we're releasing tumor-associated antigens. We're shifting, especially in tumors that are in types of cancer that are more resistant to immunotherapies, that are very, and we're trying to basically shift them into a pro-inflammatory environment. So there's a lot of work that's left to be done here exploring it, but it's a really exciting area, especially because, something from my side that we weren't really expecting to see. And now we're thinking about how can histotripsy fit on these really late stage patients, or patients with a lot of tumors or metastatic disease, as part of a therapy, or maybe repeated therapies combined with, not just the chemotherapy, but, some of these other emerging immunotherapy drugs. So it's going to be a lot of active research in the coming years in that space.
So it's fascinating to me, each of the three of you, while this isn't your primary focus on the immune system response per se, each one of you, the area you're working in, are definitely interacting with that, and it's an integrated part of what you're addressing, different types of cancers, different therapeutic approaches, et cetera. So I think it speaks to that question that was brought up. Before we go on, there are a couple of questions from the audience, and one is a follow-on to the questions you just addressed, and it's from Thomas Leitch, and let me just read it to you. He says, we've seen some exciting clinical results with CAR T-cell therapies. However, there are still some patients, particularly people who have solid tumors, that do not respond. Are there particular therapies you can envision coupling with a CAR T to get at these multiple relapse refractory patients who don't respond to CAR T alone?
I think that is that for Jenny, that question?
That's for any of you, excuse me. Anybody can address that.
Oh, sorry. Yeah, so I guess for immune therapies, So I think a big problem is that we don't quite understand completely how they interact with a lot of the standard therapies that are given to patients. And so I actually think a lot more research needs to be done, because a lot of these patients who are receiving these types of immunotherapies have often received rounds of other therapies. In the case of glioblastoma, they were receiving radiation therapy, which we know has immune consequences. They've received surgery, which causes a lot of changes to the site of the tumor, as well as they've received rounds of chemotherapy, as well as things that change things like the blood vasculature, which we know is extremely important for immune cells getting to the site of the tumor. So I think one thing that we really should focus on and understand is how a lot of these new therapies that we are putting into place, we often test them in a scenario where we haven't always given all of the other therapies that a patient is normally getting. And I think that understanding how these things interact will help us to understand how we may or may not be able to better treat patients by maybe changing a dosing strategy on something that's standard, or maybe replacing a standard therapy with something that could be more beneficial. And I do think that there's things that can be coupled with immunotherapies that a lot of clinicians do standard, including things like, and things that can be detrimental as well. But when you're talking about the care of a patient, it's so complicated because you're really managing a lot of symptoms, especially in glioblastoma, as well as trying to treat the tumor itself. And so I believe that there are a lot out there, but since it's such a new area, we're still really trying to understand, one, how these therapies really work and how can we get them to work better in patients alone, and then also how they interact with what these patients standardly get in the clinic. And so I wish I had a more direct answer to a specific therapy, but it's definitely something that we're working on.
Yeah, I just read recently an article where they actually say they link the success of these CAR-T therapies with the type of mutations that they found in the tumors. So for some of them, it seems that with certain genomic background, the therapeutics is less efficient. So I think there's a lot that we still need to learn. We don't know, and this is a common trend in pretty much all therapeutic approaches that we have to cancer for a really long time. I mean, somehow, two patients with the same type of cancer, the drug works in one, it doesn't work on the other one, and there's a lot of uncertainty of why that happens. It seems that in the particular case of CAR-T, the genetic background of the tumor is quite relevant for the success of the therapy.
Thank you. Jenny, this next question is directed specifically to you. It's from Greg Feldman. He's asking, is the lack of progress with brain cancer due to large pharma and other research funders not putting money behind research due to the relatively fewer cases of brain cancer versus other cancer types? If so, what can be done to turn this around, if anything?
Yeah, so I actually think, there is quite a bit of, large funder research in glioblastoma. There are very generous private foundations as well as government organizations that fund glioblastoma research. It's been an area of interest for a long time. So it's actually, I think, quite well funded. I think the difficulty is that it's so difficult to treat. And one of the things that we study that makes it particularly difficult is it's so invasive. And these cells migrate and move throughout the brain. And so when patients come in, that's already happened. And so getting every single one of those cells out of the patient can be very, very difficult and makes it incredibly difficult to treat. So I don't think that there's necessarily a lack of kind of interest and funding behind this. I mean, I can always advocate for more funding for glioblastoma research for personal reasons for my group, but I think that really it's just an extremely challenging disease to treat. And there are some great people working on this disease. And I think that every new thing we learn about it presents a new challenge. And so, but I do think that there have been a lot of new drugs tried and a lot of new things. So I think it just takes new innovative approaches. And I do think that's where the funding is channeling now, is trying to do really different things for this disease.
Okay, thank you, Jenny. Eli, this question for you from Kinsley Tate. Kinsley asks, are there negative impacts of using Hyssotripsy to open the blood-brain barrier? And how would this affect brain cancer treatment?
Yeah, that's a good question. So focused ultrasound in general is being developed in multiple different ways. So there's Histotripsy, which I talked about, which uses basically a non-thermal direct ablation to directly destroy the tissue we target. And this could be used for any type of solid tumor that we're developing this for. There's also other uses of focused ultrasound. So you can use it for a thermal ablation, and you can also use it in combination with microbubble contrast agents that are often used for ultrasound imaging. And that's what's typically being developed right now for reversibly opening up the blood-brain barrier. So there's a lot of work, and I know Jenny's group is working with some others on this as well, on the opening up the blood-brain barrier. And this is, like I said, a reversible effect. So with histotripsy, we think, or I should say in general, with focused ultrasound and brain tumors, it's a new area for us. A lot of our work right now is developing the transducers to do full transcranial ablation. And we do have some studies that we're going to be doing our first treatments of brain tumors in dogs through the partnership with our vet school here. So we're exploring this really early stage for histotripsy, but there we're not looking as much in the blood-brain barrier. It's more for bulk tumor ablation. And I do think because of the limitations with something like glioblastoma that Jenny was speaking to there, it's going to ultimately need to be a combination with some of these, drug delivery enhancing techniques, like opening the blood-brain barrier to deliver drugs in these regions selectively. And so far, there's a lot of clinical trials going on with that type of technology that seem very promising, but a little different than what my group is actively working on right now.
Yeah, you remind me, Eli, what you just said about the blood-brain barrier and answering Kinsley's question. I forgot to mention earlier, we have this wonderful collaborative relationship with the Children's National Hospital in Washington, D.C., and they also are using focused ultrasound. And one of the things they want to do is be able to selectively open the blood-brain barrier to deliver therapeutics. But of course, what you're using with focused ultrasound, the modulation, it seems it would be important to be able to open it as you want to open it. That is, not make it completely wide open to everything, but selectively modulate it, right? I guess there's opportunities to design parameters to do that in a more precise versus a very gross way.
Absolutely. And that's one of the biggest things about focused ultrasound. We have really high spatial and temporal resolution, right? So we can get millimeter, sub-millimeter resolution as far as the regions we're targeting, both in the brain and in other organs. So, and I believe that they, I haven't looked very recently on those clinical trials, but very promising results about on this, temporal side as well, about how long they can keep that barrier open. So you're not permanently disrupting this barrier and having other complications. But the safety profile has been pretty good in those early studies.
And I want to, we have another comment from Max Schulke. He asked me to speak about the clinical work and research with the vet school. And so we just mentioned that at the beginning. Any one of you can weigh in here, but of course, that's a very important part of the overall Virginia Tech Cancer Research Alliance, which includes a number of our colleagues from the vet school, and just a couple of great examples. And Eli, you're involved with some of them, of course, with the focused ultrasound, the Hyssotripsy work, and also the folks working, we talked about glioblastoma. And so Dr. Ross Meisel and his team and collaborators working with people in engineering, Dr. Davalos, Dr. Burbridge and others, and Jenny, and so forth. And there's a lot going on with studies on brain tumors in dogs, which have many characteristics similar to those in humans, and they occur spontaneously. And interestingly, since we just talked about the pediatric research at Children's National Medical Center, Dr. Ross Meisel and his group are part of a publication showing that indeed the molecular characteristics of these glioblastomas in dogs have a lot in common with those in children, in pediatric brain tumors. So this provides great opportunities for bringing together all these different groups, including the College of Veterinary Medicine folks that are right here, right now, in the Animal Cancer Care and Research Center in Roanoke and the Health Science Center as well. Okay, let me see. Let's go on to the next question to each of you, and then we'll come back to some more questions that are coming in from the audience. Carla, let's go back around to you. You talked a little bit about circadian rhythms and night shifts and so forth. So how can we harness the power of the body's intrinsic molecular checks to fight and treat breast cancer? The body's intrinsic molecular clocks, not checks. I apologize.
Yeah, it's a very good question. So I've been thinking about this question, and really, my main answer to this could simply say, okay, we just want to use the molecular clock in our favor for treatment, and that's where we want to put all our focus these days. And the reason for that is because very many of the therapeutic approaches that we see these days are, or we have identified that they are not necessarily applied at the right time of the day. So this causes two problems. One of the problems is that usually they're not effective. And the other one is that influence the quality of life of the patient. So why is this important? Well, let me give you an example. For example, let's say that you take some kind of drug that basically targets DNA replication, which is something that is common in normal cells, but is particularly downregulated in cancer cells. And we know nowadays that that particular process happens during the day. Now, if you take the drug at night, because you want to avoid side effects and you want the patient to go to sleep and have a better quality of sleep, let's say, and during those hours, and by the time that the drug is supposed to hit the target, which is in the morning, there might not be much drug left, okay? And that might result in the therapeutic not being effective. So the next step would be, for example, that we increase the dose of the therapeutic. So we have the right amount of drug for the next morning to hit the target. Well, that might be a good approach, but that influences for sure the quality of life of the patient, and the side effects build up over time. So the question is, can we just use what we know about how the metabolic processes are synchronized to specific times of the day to decide when to deliver therapeutics to make it more effective and without causing this detrimental effect on the life of the patient? So I think that's at least where we are going with our research in the lab. We want to identify those molecules, where the molecules that control cell divisions are present at what times of the day, but more importantly, to know when to target them so we can be more effective on doing it.
Thank you, Carla. And before we go on to the next question, there is a question from the audience to follow up you, Carla, from Terry Lundberg. She said she was a nurse for many years, worked 12-hour night shifts for 15 years of her nursing career, and had not worked night shift for 8 years, but then was diagnosed with breast cancer. Could there be a relation with that amount of time with circadian rhythm disruption?
Yeah, well, thank you very much for your question. Let me tell you that in Europe, it's not allowed to work night shift more than 5 years, and is a policy. We don't have that policy here yet. And in Europe, working night shift is considered potential carcinogenic. And is largely based in a study done in nurses. It's called the Nurses' Health Study, where 280,000 nurses were followed for over 40 years. We have 3 cohorts of nurses. And that's where we found that actually working more than 5 years night shift nonstop is actually one of the risk for developing breast cancer. There's another point. There are some individuals that actually work night shift for few days during the week, and then they switch to another shift during the rest of the week. So they kind of keep going back and forth. That is even worse for those individuals. They actually develop diseases like prostate cancer or breast cancer earlier. Yeah, take home message is, yes, it's very likely that a disease that end up a job that end up disrupting your circadian rhythm will have consequences, because your rhythm is associated or is literally entangled with your normal physiology. And if you're doing things at the wrong time of the day, working at night when you're supposed to be sleeping, your physiology is going to be all altered.
Thank you, Carla. Eli, there's another question from the audience from Skier Schulke. He asks, do you see HIFU and H-Fire to be competitive or complementary technologies? And perhaps for the rest of the audience, you can explain to them what H-Fire and HIFU are, Eli.
Yeah, no, I think that's always a question that comes up whenever you're in a department that's developing, multi types of technology. So HIFU is high intensity focused ultrasound, which typically refers to thermal ablation. And so in a sense, they're both potential options that we would use for treating a tumor. And H-Fire is, just like IRE is irreversible electroporation, and H-Fire is kind of the IRE 2.0 that's being developed here. And that's using high frequency electrical fields. And one of the, I would say both Hyssotripsy and H-Fire, the benefits we see are that they're non-thermal. So we see some of the same tissue selective effects where we're able to treat tumors in places that are typically not operable, and we can't do ablation in those areas. So I view them not necessarily as competitors on our side because we're on the academic side, and we ultimately need as many weapons we can get to fight cancers, especially some of the recent work they've done with H-fire that shows potential cell selectivity is very unique. And I think there's some applications where that's going to be extremely important for very diffuse tumors. So that's more of a question for our clinical colleagues. Our job is to kind of give them the best devices and let them pick and choose. I think from Hyssotripsy, we do think some of the non-invasive aspects in certain applications, ideally having portable that we can maybe get out of the hospital setting and have this be more of an outpatient procedure is really our long-term goal. But hopefully they're synergistic, especially when you think about some of the things we were talking about with immune response, comparing against different ablation modalities. That's really something we want to do here to make sure we can see, is there a specific tumor type that's responding to one better than the other? Can we use them synergistically? It's really too early, at least on the Histotripsy side, because we haven't advanced into the clinic in a lot of tumor types to really have that data to really see which is going to perform best in each cancer type.
Thank you, Eli. Jenny, next question for you. You had talked earlier about movement of fluid in tumors, and such as in the brain with brain cancer. And the question is, are you or your colleagues trying to develop ways to kind of freeze these tumor cells in their tracks? I mean, it sounds like from what you said about the surgeries where you can't really get it all with a malignant brain tumor usually, and the cells have migrated to other sites. So is there a way to stop them from migrating, or are there people trying to figure that out, and what are the chances?
Yeah, so there's been several therapies that have tried in clinical trials to stop the cells from invading further. Again, a difficulty is that you're often treating these tumors once they've invaded. And so there's a lot of things out there right now in clinical trials for glioblastoma and for other diseases that we believe could work very well to prevent further invasion of these tumor cells, which could really help for any sort of follow-up treatment. Because if you're really just trying to get one tumor, it's a lot easier than, a multifocal or a tumor that has multiple locations throughout the brain, which can be very bad for quality of life for patients as well. One thing that we're actually trying to do is just to try to find these cells. And that's, I think, the most important thing, because if you can find them, then you can use other modalities to target them, such as some of the stuff that Eli has spoken about, as well as, opening the blood-brain barrier and getting drugs to specific locations. And so one thing that we believe we're able to do through development of MRI imaging methods is to actually image fluid flow and then use that to identify these invasive regions of the tumors in a more robust way than just standard imaging techniques. And so that's something that we're working on both clinically as well as preclinically to actually see if we can find these cells and then actually get the drugs to these cells so that we make sure that we're treating them. And so kind of using complementary methods in that way to get to them and then worrying about stopping them, because I think what's so hard is actually finding those cells and getting therapies to them in the first place, whether those therapies stop them invading further or kill them, which is what we ultimately want.
Great, thanks, Jenny. Next question before I go back to the questions coming in, another one that I prepared for y'all, is for all of you to take, really. And the comment I'm making here is Virginia Tech has a lot of people doing cancer research. We've talked about that. We have some of the great ones here. But we have some differences here. Unlike a lot of cancer centers around the country, our cancer researchers are really distributed, and as I mentioned at the outset, across different colleges, institutes, different locations. You're working on different approaches using biological, chemical, engineering, computational approaches, etc. So my question to all of you, do you see this as a strength or as a weakness for Virginia Tech for cancer research, and why? Any one of you?
Well, I personally think it's a strength. I really like the fact that we are all focusing, tackling the disease, and everybody does it from its own angle, right? And it's not just a unique angle; it's very different. I mean, I don't really care if technology works or not. I mean, it's just as long as we cure cancer, I'm totally okay. It's just if that solves the problem, get rid of the tumor, I'm totally cool. And it's really fantastic to have colleagues that have completely different approaches to actually tackle the problem of cancer. So I personally think it's a strength, but certainly being sparse all over campus, I don't know. I don't see that as a particular weakness in any way. I mean, I have colleagues in Asia and colleagues in Europe, So, I mean, I don't know. What do you guys think?
Yeah, Eli, Jenny, what do you think about that?
Yeah, I mean, I guess I can make a couple comments. I obviously agree with that. And I think this always has the potential to go either way. And I think one of the reasons it acts as a strength for us is that we're not really siloed. I think that's kind of the culture here. And we have this very, cross-department, cross-college, interdisciplinary environment. I felt that right away when I came in here. And that was really important to me. That's one of the reasons that I liked Virginia Tech, is not only, having great clinical collaborators here, as well as external clinical collaborators that I work with, but having the colleagues at the veterinary hospital, especially for doing device-related work and trying to study these in large animal models. But even within our lab, we need to have electrical engineers, mechanical engineers. We need to be able to work closely with clinicians to tell us, what are you trying to treat? What are the risks for the device, or where does this fit? We already mentioned the immunologist studying the cancer basic biology. So there's this whole range that comes together on every project, and the ability for students in my lab, many of them have co-advisors, and we work very closely together. So I think the fact that it's a strength for us is mostly due to the culture we have around and the way it works out between working together and really pushing that. And so, sorry, I didn't know. So ultimately, I think that's working. I mean, Jenny and I have a few projects that we were able to get started very quickly together. But like I said, it's not limited to our department or even Blacksburg versus Roanoke. We have a lot of medical students who are working across both campuses. So I think it's really working in our favor and allowing us to kind of take everybody's expertise and kind of throw all of these different different ideas at the problem from different perspectives.
Jenny?
I think also just by its nature, because I think we are a group of people who have all been interested in working on cancer, but then we've kind of come together from a lot of different angles. I think it just leads to a lot more creativity that, I don't think any two of us have the same training, as opposed to, I think if you're starting to build something from the ground up, like a concerted center, you might really focus in on one area. We kind of have, a lot of different areas because we come from all sorts of different departments and different expertise and different backgrounds. So I think that leads to a lot more collaborative creativity and flexibility and just innovation that maybe isn't traditional. So I think that's a big advantage, though it can be challenging just communications wise and just figuring out where these things are.
But we don't need traditional anymore anyway. I mean, we've been doing traditional cancer research for
right.
Really
it's not working.
And we definitely need something that is completely out of the box. So I think that's why bringing all this interdisciplinary research that we have on campus is so good. Plus we get to learn from.
And our students are great and they love to work across boundaries.
Yep. They are fearless.
Yes, they are.
And related to that, before I get to the next question, I'm going to read a comment from Frank. He says, this research makes me proud to be a Hokie. Ut prosum. Thanks for having the broadcast. I hope you all be doing more. So I think that speaks to you all and your teams and your students you mentioned, et cetera. There's a question from the audience from Dr. Penberthy that actually touches on something that each of you mentioned a little bit at the beginning. We want to come back to it here, maybe a little more depth. He says, can any of you describe the effort at Virginia Tech regarding test development and or analyzing genetic information for the purpose of developing targeted therapies? So could any of you want to share with Dr. Penberthy, what's going on either in your own labs or other labs, that are taking advantage of this? Carla, you had talked earlier about genotyping tumors and breast cancer, but anyway, go ahead.
We have done something like that, but really focus on clock genes, right? So I just want to take my answer with a grain of salt. So this is, so the idea that we have in the lab is that the regulation of cell proliferation, or the fact that the cell divides without any control, is due to the lack of control, timely control of when to divide, okay? So the cell should divide at a specific time of the day and only once, and we think that when that signal is deregulated is when things happen. Anyway, to make a long story short, we went back and checked for those clock genes, the genes that control the timing of division, and we found out that in certain tumors, especially in breast tumors, there is a number of mutations that occurs in a particular region of these clock genes that are that they directly impact the function of the clock system, and because of that, we think that the cell gets the wrong signal all the time. Now, we really focus only on looking at genotyping and clock genes simply because it's what my lab is all about. But we're thinking on using this as a marker for specific therapeutics as well. I don't know if my colleagues have done any particular genetic background and their studies and their research.
Yeah, I mean, it's not an area that we're actively involved in. There's a lot of interest right now on our side of developing Hyssotripsy enhanced liquid biopsies to kind of promote some of the liquid biopsies, because we've seen that you can actually release DNA and RNA. Ironically, we have a large project in our lab focused in environmental conservation where we're developing focused ultrasound technologies for rapid DNA extraction from timber and leaves and other biological samples for other applications that we think is relevant to lay some of the groundwork for this. But my hope is that some of that can, as we start to see, emergence of different immunotherapies, that maybe a Hyssotripsy enhanced liquid biopsy could, we do a primary treatment, and from that, those markers, we could determine, what type of immunotherapy to give for those patients. So it's not really work we're doing, but I think it's an area that's going to really improve decision-making going forward. And we have some colleagues who are working on that here, but I don't want to speak to their work and have them send me some mean emails when I describe it wrong. So I'll stop there.
Okay. So Eli, we can stay with you for just a second. So you told us a bit about Hyssotripsy and destroying tumors, but a question that comes up, and I think for a lot of folks, there's curiosity about this. People hear about different therapies for cancer using powerful sources of energy, radiation beams, etc. And there's always this fine-edged sword, right, between damaging what you want to damage, the cancer cells in the tumor, and not damaging the surrounding normal, healthy tissue, whether it's the brain or the breast or whatever it might be. So can you tell us a little bit more with these technologies that you use? How is it that they don't or shouldn't damage surrounding healthy tissue? Where does the specificity come from?
Yeah, no, that's a great question. It really gets at the core of what we do when we're developing devices. So I'll say first off, comparing this to like radiation oncology is significantly different because Hyssotripsy involves threshold phenomena. So it really depends on this really distinct cavitation threshold to generate these microbubbles. So along the tissue path, we're generating no cavitation. It's only right at the focus. Whereas with a lot of targeted radiation, you're basically getting a higher dose at the center, but there's still some radiation dose along that path. So it's a matter of managing the dose for getting the most toxic effects to kill the tumor at the focus. But as far as overall precision and tissue selectivity, there's really a few ways that we go about this as we kind of walk through our device development or technology development for different pulsing parameters. So the first one is that we're really in the device space. So we custom design all of our systems for specific tumor types, for specific patient populations. So that's the size, geometry of our transducers, looking at imaging data, looking at where the tumor average size location are, and customizing the devices so that we can be highly precise and localized to that tissue of interest. Second, and probably most importantly, is that all of our techniques are image guided by real-time ultrasound imaging, or in the case of the brain, we're using MR guided treatments. And this allows us to have, with the therapy, millimeter precision guided by real-time imaging. So the clinician is looking at the image, mapping out the region that they want to treat, and then turning on the Hyssotripsy, seeing in real time where they're treating and watching the tissue effects as the tissue is ablated into this acellular debris. And then we have robotic micropositioners that are allowing us to uniformly treat that targeted volume. And because it's a non-thermal ablation, we can kind of carve out arbitrarily sized and shaped volume. So that's really the main way in which we precisely ablate the tissue of interest. Beyond this, though, a large portion of my former work has been looking at differences between different tissue types with Hyssotripsy. And due to this non-thermal effect, we've seen that tissue properties, particularly the tissue mechanical properties, are, have a significant impact on the ablation. So stiffer tissues are more resistant to damage. So what we've actually been able to do with this is tailor our parameters so we can get tissue selective ablation where we completely ablate the tumors while preserving large blood vessels, bile ducts, and other critical structures that are inherently stiffer. So you have a lot of tumors that are in high-risk patients that they really can't get surgery or current ablation because the thermal ablation will, potentially damage these structures. So we think this can be a potential game changer, really taking Hyssotripsy into those applications where we can get a selective ablation, even if you have a primary bile duct right through the ablation zone. So that's really an exciting area. And we've had a lot of data on that now, including our recent work starting with bone tumors to selectively ablate the tumor while preserving that, the healthy bone that's remaining there. That's kind of the core as far as tissue selectivity. I will say, when Jenny's talking earlier about brain cancer, or Carla's talking about breast cancer, in these really late stage patients where you have infiltrating cells and you maybe not be able to image them or see exactly where a larger tumor is. So most of what I've talked about so far is bulk tumor ablation. So we have separate projects that are going right now is developing what we call nanoparticle guided histotripsy. And this uses acoustically active nanoparticles that are selectively targeted to cancer cells. We're starting, Jenny and I, along with Coy Allen at the vet school, have a grant developing this starting for metastatic breast cancer, and we're interested in brain cancer as well. And the way this works is they selectively target these cells, and then we apply focused ultrasound at a lower pressure, and you only blow up the tumor cells where these particles are located. So that's earlier, and we hope this can get us to really the cell selectivity level as opposed to this image-guided tissue selective level, and really get us to more of these really difficult to treat diffuse late-stage tumors.
Yeah, thanks, Eli. Based on what you said, I don't know, I may be getting into risky ground here. It sounds like I heard potential collaboration between you and Jenny. When you talk about the mechanical properties and the stiffness of the tumor, I would imagine interstitial flow, Jenny, is directly or at least related in some way functionally to those mechanical properties of the tissue around the tumor as well. Would that be correct?
Yeah, they're closely related, and kind of throwing back to my more engineering roots, you have kind of the solid mechanics, which is what Eli was talking about, with the fluid mechanics, which is what we really look at. So we know things like, and this happens in other tissues, that if you have increased fluid flow, it can lead to things like fibrosis. So in the liver, this happens, but it also happens in breast tissue and can lead to stiffening of the tissues. And that can happen in a lot of diseases, not just in cancers. And so that's one of the things that we know is really related. And so Eli and I have talked a lot about how we can, kind of use his technology to change fluid flow, and then vice versa, how just understanding how his technology might change fluid flow and what is it doing to the tissue that can cause that. And that's where we have a real strength at Virginia Tech, because we can really pull upon, some really advanced computational work going on across many departments in the more basic, not necessarily in the, medical research, but people who really understand how these types of really complex mechanics interact. And so it's really nice to see how you can translate, this more, I guess, basic research and in more, dry research or computational research into a clinical setting. And I think that's where we might see a lot of benefits in the future, is by taking those kind of broad strokes approaches.
Great, thank you. A little bit of time left, not much, but Carla, there's a question from the audience. Eileen Caridad Suarez would like to ask you, how does the hormonal fluctuations during a menstrual cycle affect the patient's immune response and breast cancer in general?
Yeah, there's a theory non-proven yet, but it has been out there, that is really the dysregulation of the estrogen levels, that they are caused by either the accumulation of estradiol because it's an unwanted phenomenon, or because they are artificially elevated by treatment that cause the signal for division. Let me put it in this way. Estrogen is basically, imagine that estrogen is like a signal that goes to the cell and tells the cell to divide. Now, under normal conditions, we get the signal and it's transient. It happens, it's a peak and goes down. In cancer patients, we've seen that they have a very steep increase and remains high. And that means that continuously receive the signal for division when they shouldn't be receiving that signal for division. So the point is here, if it goes up and down, then it can be controlled. And if it goes up and stay up is what it causes problems. And that happens usually in treatments in women with postmenopausic treatments, with hormone replacement therapy that was associated with increased number of breast cancer cases. In the regular cycle, because it's just transient, there is no any potential problems or consequences.
Thank you, Carla. So we're down to the last minute or so here. So I'm going to, first of all, a question comes in from Max Schulke. He says, that I mentioned, Children's National Hospital Collaboration. He asked if there are any other centers we can mention. Well, as my colleagues know, there's probably about 50 I could mention, at least, both within the United States and overseas as well. Actually, it's interesting you asked that question, Max. We just collected a lot of that information for our investigators, and whether it's MD Anderson or Memorial Sloan Kettering or Johns Hopkins or Duke University Cancer Center, Wake Forest University, very good collaborators and colleagues that were folks here. There are a number of other very important cancer centers and universities that our investigators have great collaborations with. Since we've sort of hit the time, I'm just going to end it with a comment from one of our guests. Somebody's asked the question, Dr. Penberthy, and he says he'd like to comment. He says he's really glad to see Virginia Tech in this space. By the way, he's a proud Virginia Tech electrical engineer grad, BSEE, and has been practicing radiation oncology for the last 22 years. So the discussion makes him really proud to be a Hokie, and he'll likely email some of you to continue the conversation. This really is a great time to be in oncology. The future is bright and the outlook is better than ever for patients everywhere. And I think that has to do as much with these wonderful clinicians we have in the area and all over the country and the world, and the researchers sitting right here, some of whom you've heard from, and it sure makes me proud to be a Hokie as well, to have colleagues like this. So without further ado, being that we're on the 8 o'clock hour, I'd like to thank very much the entire panel. Carla, Eli, Jenny, it's been wonderful, and I'd like to thank our entire audience for the conversation this evening. We hope you all enjoyed it. Have a wonderful evening. Thank you.